I think, perfect. Got the meeting recording. So first off, I'll introduce myself. My name is Jackson Burkholder. I'm on the leadership team of ICANN. Do you want to introduce yourself, Eddie?
Yeah, for sure. Well, I'm Eddie Mendoza. Right now, I'm studying in Montpellier University in France. Yeah, so I am a supporter of the ICANN activities. I'm very happy to have you here, Dr. Kevin. Thank you so much for accepting.
Yep. Both happy to have you here. So with on that note, I'll introduce our speaker. So our speaker today is Dr. Kevin Hultine from the Desert Botanic Gardens in Phoenix, Arizona. He got his PhD from the University of Arizona and is currently the research director at the Desert Botanic Gardens, studying how plants cope with extreme heat and drought stress in deserts and how they respond to changing climates. So on that note, I'll mute myself here and I will let Dr. Hultine take it away.
Wow, well, thank you so much. It's great to be here, and I'm really impressed. I think by my count, there are at least in this group, there's at least 7 countries represented, and I could be wrong. I'm going to share my screen, see if this works. And can you all see the screen?
Looks good.
Great, great. Well, first, just a little bit of background about this work and just sort of a disclaimer in the title. In fact, if I go back one, the title suggests that we know in the ways that giant cacti impact the ecohydrological processes in these desert ecosystems. The truth is, we really don't know how they impact the ecohydrology and hydrological cycle. We have some pretty good ideas based on data we've collected over the years. So I'm going to basically present some ideas of how we think giant cacti are impacting the hydrological cycle and ecohydrological processes more broadly in these dryland ecosystems. First, I want to just introduce some co-authors to this work who've been really instrumental in collecting the data that I'll be showing today, and that includes Shannon Albecki, Alberto Borges, Newton Tran, and David Williams. I'll get started from there. So first things first, I think a lot of you already know this, is that these dryland ecosystems are very definitively shaped by infrequent rain pulses and residual water storage. But typically we think of water storage in the soil itself. However, when you think about giant cacti where there's water storage in the stems instead of the water, what we think is that the basic rules that govern ecological theory are strictly based on plant access to soil water storage, not on water stored by vegetation. And that water storage component in these giant succulents itself can really impact how water moves through an ecosystem, how it's converted to productivity. One way to look at this is through this thing called the pulse reserve model. This was originally put together in the 1970s, and then it was updated in the early 2000s. And if you follow this, this model from left to right, starting with something like a typical creosote shrub, you'd say a pulse of water, in this case, a rain pulse, would then lead to soil water storage, and that water would be taken up by roots and immediately turned in to plant products through gas exchange, where water is lost through transpiration, but photosynthetics are then acquired, and that leaves over time this reserve, which is ultimately turns into seeds, roots, and stems, etc. But what that model doesn't really show is how something, if you have giant succulents in the landscape, you actually have a disconnect between root water uptake and plant gas exchange. And there's this important intermediate step in which water is first stored in stems before it is taken up as gas exchange. And that storage component potentially delays that reserve for a much longer period. And so the reserve of seeds, roots, and stems and may actually may not actually happen until, in some cases, years after an individual pulse event, depending on the size of the event and the time of the year that it actually occurs. So that leads us to thinking about then more broadly about how that storage component could impact the ecohydrology of these desert ecosystems. And to do that, we have to start with sort of the classic CAM biology trait syndrome. So there are clear traits that CAM plants have that are very important to make them very unique compared to something like a C3 plant. So first of all, of course, it's just CAM metabolism itself. As we know, CAM plants take up water or take up CO2 at night. They lose water primarily during the evening as well. And that material then is stored as malate, which is converted to malic acid. Eventually, it is decarboxylated back to CO2 during the day, where it's fixed by C3 enzymes. So that's one way. This is a very high water use efficient pathway. It is also very costly in terms of the metabolism itself. So it's a trade-off. But that CAM metabolism typically also in strong CAM plants is part of that, a clear trait where there's a lot of stem succulents or leaf succulents at least. And that leads to stem water storage. And this is something I'm going to talk about a lot, the impact of stem water storage and what that means in terms of how water is then converted into various products like seeds and stems, et cetera, and roots. The other thing, we typically think of CAM trait syndrome as plants that have very shallow roots, and that's certainly true with almost all cacti plants. But I'm going to put an asterisk on this one because these giant succulents, like giant saguaro, for example, have rooting depths that might be surprisingly deeper than one might think, and that impacts their capacity to govern soil water storage because they can collect water from multiple depths. And I'm gonna, like I said, I'm putting an asterisk on this one because I'm gonna go into this a bit in a few minutes. And then finally, as I mentioned before, this is going to be a common theme throughout the talk, and that's this decoupling between soil water uptake and gas exchange. As I showed in that previous model, this is a really important point that we seem to forget about, that gas exchange and root water uptake aren't necessarily in phase. And because of that, the plants could take up a lot of water regardless of what the environmental conditions are, if there's water in the soil to begin with. I'll discuss quite a bit what that might mean in terms of total ecosystem water balance. Okay, so that's a CAM trait syndrome. And based on what we know about how these giant succulents operate, we could put forth this very basic overarching hypothesis, and very broad hypothesis that just simply states that the present of giant cacti fundamentally alter soil water balance and dryland ecosystems in ways that are not yet captured in extant model theory. So I'm going to describe a little bit how we think, at least, that sort of the classic models that describes water movement through these dryland ecosystems may actually not be entirely relevant to systems dominated by giant cacti plants. Okay, so here's a little thought exercise. This is a photograph of several saguaros growing under a mesquite tree. This is just north of Tucson, north of the Catalina Mountains. And if you look at all these small saguaros that are clearly being facilitated by this nurse plant, but you think about how much water do you think is actually stored in these plants that's actually removed from the soil water itself, which could impact, of course, the function of this mesquite tree over time and then surrounding biota as well. So if you take a quick look, make some guesses, we did a back of the envelope calculation and decided we came up with a number of about 475 kilograms or 125 gallons of water stored in all of these small saguaro plants. And that equates to about 20 millimeters of depth of soil water, which may not seem like a lot, but that's about 10% of the annual rainfall on this site. So you can imagine that that 10%, when you think about this as a water limited system, could have pretty profound impacts on how water moves through the system and how the rest of the ecosystem actually is able to use water, or in this case, may not be able to use water because it is taken up now by saguaros, which has a lot of storage. We can think about then what is the water storage? What is the capacity of cactus plants? As you all know, what makes cacti really interesting is they come in all shapes and sizes. And this is just a graph showing a relationship between a number of cacti species in terms of the water storage in grams per centimeter of stem length versus their volume to surface area ratio in cubic centimeters to square centimeters. So something like a pencil cholla would fall down here, very low VS and very low water storage. But if you wonder where something like a giant saguaro falls on this graph, actually it doesn't fall in the graph at all. It'd be somewhere up here, probably somewhere near the ceiling of where you're sitting right now. So the key point is that total soil stem water storage could range by a couple of orders of magnitude across different cacti taxa, with some giant cacti being the real winners of total storage capacity. And so again, just giving you an idea what the range could be across these different species. So a couple species that we've been studying quite a bit, and it's really interesting in the Sonoran Desert, they have very overlapping distributions. So one is giant saguaro, which grows, it's very prominent in the northern Sonoran Desert throughout much of the Sonoran region of Arizona, as well as organ pipe cactus that also has, again, grows throughout the Sonoran Desert, also grows quite a bit in southern Baja. The interesting thing is that they have, of course, different growth rates, they have different life forms, they have different storage capacities, and they have different photosynthetic expression through time that I'm going to show you in a minute. So the key point is that even though they are overlapping, they could have very different impacts on the ecohydrology of the ecosystems in which they occur in. So just one quick comparison then, what is the storage capacity? How do they compare over time? One way we can do that is measure stem diameters of various plants growing in the same location and simultaneously measure the stem water content. So the left panel here shows the relationship between stem diameter at a single location in Phoenix, Arizona, and stem water content. And you can see how closely related water content is to stem diameter at the site. But it's also quite remarkable just how much water is being stored. So when the plants are most turgid, you can see that total stem water content is actually over 90% in the stem. Likewise, in organ pipe, the right panel there, kind of a similar relationship with stem diameter. But we could use this now this information to calculate the amount of soil, stem water storage that's occurring over time. So this is a couple of graphs showing changes in total stem water storage in grams per, again, centimeter of stem length. And so focusing just on the upper panel first, you can see that over this about a 3.5 year period starting September 1, 2014, these are measurements taken about every 2 weeks on 15 saguaro and 15 organ pipe plants. And first things first, you can see that saguaro has stores about 6 to 7 times more water than organ pipe relative to stem length. The other thing is you can see how dynamic these values are. So in saguaro, it changes from about 700 grams to during the driest periods before the monsoon below 500 grams. Likewise, in organ pipe, you can see it can range from over 100 grams per centimeter down to about 50 grams per centimeter. So again, so measured about over 45 months, organ pipe loses a higher percentage of stored water during the dry season. And that's what this lower panel is showing. So again, the x-axis is days following this September 1st, kind of the end of the monsoon in 2014. And the y-axis is the percent of maximum water storage. So the maximum being there around day 900 is very wet during that period. But the interesting take-home message here is you can see that organ pipe has sort of a riskier strategy where it's willing to exhaust a much larger percentage of its stored water. In fact, you can see that total water storage during the height of the pre-monsoon, during the driest time of the year, drops below 40% of its maximum, where in saguaros, it never really drops below 60% of its maximum. So 2 different water use strategies. But what's really interesting to me is if you look at this dry down period that takes us from spring into early summer before the monsoon, and the actual rate of water loss then is pretty similar between saguaros and organ pipes. So what this is showing us is that despite the fact that organ pipe loses a higher percentage of its stored water, it's maintaining a similar apparent transpiration rate. So again, it's a riskier strategy. It's moving water much more quickly than saguaros do. So if you think about the residence time of water in the stem, organ pipe has a much lower residence time. And so that reserve then, that length of time that reserve that we talked about in that pulse reserve model is going to be much shorter in organ pipes relative to saguaros. There are some other traits too that we can relate then to stem water storage, and that is just how these stems are built. And this is a model that was developed by a colleague of mine, Alberto Burques. He's at UNAM in Hermosillo in Sonora, Mexico. And this just demonstrates how the number of ribs allows plants to store water and to shrink and swell, kind of like an accordion. So you can see that the more ribs you have, the more that you could basically blow up and store water. And so, and indeed, we found that in saguaros, in the wetter locations, they typically have more ribs and they have more of a expansion during the monsoon season or during a wet winter than plants growing in drier locations. Now, another obvious benefit to having more ribs, you produce more aerials. More aerials mean more places to produce flower buds. So if you could avoid, if you could actually have the resources to build more of these outer ribs, there's a dual benefit in doing so. And so this just gives an example how the mechanisms by which saguaros, and to a somewhat similar extent, organ pipe and other cacti species can shrink and swell. Okay, so this is we talked about the CAM trait syndrome. That was basically a overview of water storage. But what about CAM photosynthesis itself? So we did a little project where we measured the photosynthetic rates of these small cactus plants. We actually measured several species, but I'm going to focus on two, saguaro and organ pipe again. So we put them in this small chamber with fans, and then we actually measured the changes in CO2 concentration and water vapor over time. And we did this through over a diurnal period to get an idea of how much photosynthesis these plants are actually doing during the springtime and in the summertime relative to water loss. So this is next a couple panels showing the total net CO2 flux of plants over a diurnal period. So that upper panel is taken in April from 5 organ pipe plants and 5 sorrel plants. D there at the top of the panel represents vapor pressure deficit at night, or particularly early in the morning. So vapor pressure deficit is really the driving gradient that drives transpirational water loss from plant tissues. And so the higher that number is, the drier the air, the hotter the air is, and the higher the evaporative demand is. So in April, VPD was around 1, and that should be actually kilopascals, not megapascals. And so, and that number is fairly low for a desert ecosystem. But in that upper panel, you can see that organ pipe maintained much higher photosynthetic rates than saguaro did. The other thing is that these plants are as cammy as CAM plants can get. So many plants do what is called 4-phase CAM. So you know CAM plants, as I mentioned earlier, they take up CO2 at night, and then it gets recarboxylated and fixed into C3 sugars during the day. But a lot of plants, a lot of CAM plants, when they're well watered like these potted saguaros and organ pipes were, will actually take up CO2 as well in the early morning, in the late afternoon. So then you would see a spike, and see around here, if you see my pointer, in the early morning and possibly in the late afternoon before it gets dark, of CO2 uptake. But we did not see that at all in these plants. Instead, we saw a lot of what we call a net efflux. And this is basically because there's two things going on. There's a lot of mitochondrial respiration, but it also shows evidence that not all of that CO2 taken up at night was actually ultimately fixed in the 3 carbon sugars and was instead leaked out of the plant. And so if you look at over this 24-hour period, the net CO2 uptake in saguaros was basically near 0. It was a little bit higher in organ pipe plants. In August then, so VPD now is above 2 kilopascals, so there's a higher evaporative demand. And typically this means that plants will have a lower photosynthetic rate because the stomates begin to close when it gets dry, when the atmosphere gets drier. But what you see is now saguaros have a much higher photosynthetic rate that matches that of organ pipe. And so one difference here between the two plants, organ pipes pretty much grow year round, except maybe in the winter. Typically saguaros only grow from about late June through October. And so you can see there's clear evidence of a source sink relationship here, where as they are growing, they're now taking up more CO2. But again, not much happening during the day. There's more leakage. And so in April anyway, that organ pipe had about a 2 times higher nocturnal flux, but that flux rate then disappears. That difference between the two plants disappears in August. So the take-home message here is that there are different photosynthetic processes that play into the amount of water that gets taken up by the plant and stored. So this is just looking at those two periods, April and August, and this is looking at that early morning period. So starting on the upper left panel there in April, that A net, that's a net photosynthetic rate, again, early in the morning, and you can see that 2 times higher net photosynthetic rate that we showed earlier, organ pipe having these much higher photosynthetic rates. But you look at the total transpirational water loss, and you can see that it's really more like 3 times higher in organ pipes. So again, feeds into that idea that the residence time of water in the stem is much lower. That bottom panel there is stomatal conductance. And you can see stomatal conductance is again about 3 times higher at night in organ pipe versus saguaro. But those differences largely disappear in August. And in fact, if anything, they're a little bit higher in saguaros in August. So again, no contrast, but an important point here is when we think about cacti plants or cam plants, especially these kinds of cam plants are doing what we call two-phase cam, could have exceptionally high water use efficiencies, which is defined as net photosynthesis versus amount of stomatal conductance. And in this case, these plants are operating with a water use efficiency of about 100 to 300 micromoles of CO2 uptake per mole of water loss. Now, compared to something like a creosote brush, that's probably about 10 to 30 times higher. So these are among the highest water use efficient plants, really, that we know of on the planet. They're quite remarkable in that sense. But that high water use efficiency, again, feeds into their capacity to alter soil water balance. Okay, let's move on then. I mentioned earlier that I wanted to put an asterisk on rooting depths. And I'm going to explain why here. And that is, we at the Desert Botanical Garden did these experiments where we actually put these small sensors into the roots of a single saguaro plant and a single organ pipe plant. And what these sensors do is they measure the rate in which sap or water is flowing through the root itself. So you see there are 3 probes there. Those 2 outer probes just measure temperature, and the 2 in that red inner probe, it actually releases a heat pulse. And so the way this works is that this simply measures the amount of heat that's carried up and downstream from that probe, and we can convert that directly to the rate in which water is moving through either a root or a stem of a plant. So we went ahead and instrumented several roots of this saguaro plant and several roots of a nearby organ pipe plant as well. This is a pattern we found. Okay, so I'm going to actually start with the lower panel first. The lower panel is the x-axis is day of year. So this we started making measurements around March 1st, and this goes through for about 2 months into May, and this is volumetric soil water content measured at 3 different depths at 5 centimeters, 15 and 30 centimeters. And you can see that there is a pretty strong pulse event that occurred here around day 70, smaller event around near day 80. Okay, so this panel here is looking at the percentage of maximum sap velocity. So this is a rate that water is moving through that root. So let's start with this organ pipe root called OP East, and you can see how rapidly it responds to this pulse event. And so you can see that it basically reaches its maximum velocity over that 2-month period, almost immediately after this pulse event, where soil water actually increased at all 3 depths down to 30 centimeters. But you can see also how quickly it falls. And so this is indicative of really shallow rooting depths. It's really only capturing water that is maintained in the soil for a pretty short amount of time. But you can contrast that to these 2 saguaro roots, and you can see that they also had a response to that pulse event, but you can see that they are capturing water for a much longer period of time. And it really isn't until about day 100 that we see that sap velocity drop to levels that are near of what that organ pipe fell to. So what this means is that these saguaro roots are likely much deeper, and they're able to capture water across a much broader vertical range of soil depths than organ pipe is capturing. We can compare that then to what's happening in the stem. So we had these electric band dendrometers placed on the stems of these plants. So starting with saguaros, these 2 dark blue, navy blue, and light blue traces here represent dendrometers placed on the stem at 1.5 meters and 3 meters above the soil surface. And you can see that across this 2-month period, that again, the y-axis being the percent of starting stem circumference. So when we start here, we're right at 100%. And you can see by the end, it's still near 100% in the stem. So what this means, a couple of things, that is leading to the balance of water being much larger in these saguaro stems. And that's because of there's more water being taken up over a broader period of time, and that's what that middle panel is showing us. But if you go back and remember, those photosynthetic rates and water loss rates in saguaros are much lower than organ pipe. And so at the end of this period, saguaros are still maintaining about the same amount of water in the stems relative to organ pipe. These 3 organ pipe stems, in contrast, these are 3 stems of the same plant, again, same plant that we measured root sap flux. You can see that after water is uptake is dropped to near 0, that water storage in the stem begins to rapidly decrease. And again, they have a riskier water use strategy, especially in the spring. They have less water being taken up. So you can imagine then the residence time in these organ pipe stems is much lower than in these saguaro stems. I know it's a lot to capture, but we find this really, really interesting in how the water use components of these 2 species are really different. In fact, I would argue that organ pipe is almost like more like a shrub than what we think about a succulent plant. We could look at this a little bit differently as well by first comparing these diurnal sap velocity rates of a saguaro versus a typical tree. So this is looking at the sap velocity in centimeters per hour in a cottonwood tree, and this is exactly what you would figure you would see. During the daytime, you have a much higher uptake, of course. Remember, these are C3 plants. They don't have a lot of storage, so they're taking up water during the day, and that water is then being released through the leaves at the same time. There is a little bit of water loss at night. They do have nighttime transpiration. That's just to show that this is a typical C3 tree species and what it looks like. Well, this is what we found in saguaro, that one saguaro there at the Desert Botanical Garden. And this, to us, was really interesting. And so first of all, you can see that there is an uptick of water of sap velocity during the day, even though we know that the plants are losing water at night. And so what we think is going on here, in fact, I'm going to just, that's what that dendrometer looks like. And I'm just going to add this trace as well. So from the dendrometer, this is the relative change in circumference. And indeed, you see that it flattens out at night. And this is probably related to water loss in the stem. And then it goes up during the day. But it's going up during the day for two reasons. One, it's not losing water during the day. But two, you see that there's this higher flux rate during the day. And so what we think is happening is that the osmotic potential in the storage tissues is actually going up. Even though malate is being produced at night, remember that malate production is really near the surface in those green chlorenchyma tissues. And so there's a real lag time in which water then is being, when those gradients are increasing during the day. So this could just be simply a capacitance issue. And so there's a delay in which we start to see a little bit of an uptick in the roots, or it could be that the actual osmotic gradients are getting larger in the storage tissues during the day. We don't really know the answer to that. We do plan to measure osmotic potentials across this different tissues in the stem to get a better idea of what the diurnal patterns are in these osmotic gradients. But it gives you an idea then how dynamic water uptake is in a plant, as well as how that water then is propagated in terms of stem water storage throughout, not only throughout the day, but throughout different seasons for these plants. So a pretty complex system of water storage. And again, this is just looking where that storage is likely occurring. This is a cortex tissue here. And so we really want to know if there are osmotic gradients that are propagated beyond this green chlorenchyma tissue into the cortex, which is the primary storage tissue where water is moving into during the both day and night, but especially during the day. Okay, so putting all that together, and this is where admittedly that this becomes a bit speculative because we really don't have all the information yet to say that this is really happening, but this is what we think are happening here. So starting, this black line here represents what we think would be the pulse residence time of a creosote or other shrub dominated system. And then these arrows represent the extent to which both organ pipe and saguaro would impact that, in this case, is pulse residence time. And so we're pretty convinced that the residence time of water, and this is not just water in the soil and water in the stem, this is total ecosystem residence time after a pulse event. So it's kind of a little bit confusing, and it's kind of, there's a lot of complexity to this concept. So what we think is that in an organ pipe dominated system, that given that we have smaller stems, higher photosynthetic rates in organ pipe, that they would still increase the residence time relative to a creosote dominated system in the summer, but not nearly to the extent that saguaro will. Simply, again, those large stems, low photosynthetic rates, deeper roots, able to capture and hang on to water in the stem, that we think in the summertime that there would be a pretty dramatic change in the residence time of that water. And again, if it's sitting in the soil and you have a small pulse event, that water is going to be lost pretty quickly to evaporation. But the shallow roots of both organ pipe and saguaro can capture that water, bring it into the stem where it is not as quickly released through transpiration. In the wintertime, I think there'd be very similar kind of process, except if anything, in saguaros, it would be even longer. And that's again because of that slow photosynthetic rates, and water is held on to the stem for a long time. So again, this is a bit speculative. This is the hypothesis, but that's what we think is would happen. And then if we look at winter soil water storage, and how it is governed then by stem water storage, we actually don't think in the summertime that it would change very much between creosote, organ pipe, or saguaro. So that's why you just have this black line, no arrows. As far as we can tell, soil water in the summertime is going to get depleted pretty rapidly, regardless of what the composition of plant life form is on the landscape. However, in the summertime, in the wintertime, we think they would change quite dramatically, and that soil water storage would be more rapidly depleted in a system that is especially dominated by saguaros. And this goes back to the same concept. Lots of a very broad vertical rooting depth in saguaros, and all that stem water storage, and the slow release of water from the stem over time means that they can very efficiently take up soil water in ways that creosote cannot. And remember, creosote also is only taking up water after a pulse event during the daytime when there is actually a driving gradient. But remember, in these large succulent stems, the driving gradient is not the dry atmosphere, but is the stem osmotic potential itself. And so that allows the stem to increase its total capacitance of water. And finally, just a couple more concepts here. Cactus ecosystems, we think, because of all the CAM trait syndrome, that cactus would establish a higher transpiration rate to total evapotranspiration. And what this means, when you have water loss through transpiration, that water then gets turned into productivity. It produces flowers, seeds, etc., that will help basically provide the foundational support for these dryland ecosystems. So we think that over the course of an entire year, this is annual, this isn't summer and winter, this is the actual annual amount of transpiration per ET. We think that both organ pipe and especially saguaro would increase that percentage. And then finally, if we look at then net primary productivity, this is amount of either carbon uptake, net carbon uptake, or net growth. We think that in these organ pipe and saguaro dominated systems, that we would indeed get a much higher NPP to the amount of precipitation that falls on a landscape. And this is again because of all that internal water storage, diverse rooting depths, and CAM photosynthesis. Okay, just some final thoughts and conclusions. I know there's a lot of information. And again, this is a bit speculative. We still haven't done the full ecosystem analysis of how water moves through the system, but that's the next phase of what we're working on. So first of all, we think the presence of giant cacti in dryland ecosystems does indeed have profound impacts on the ecohydrology and water cycling of these dryland ecosystems in ways that haven't really been quantified to date. And so the mechanisms by which they would impact the ecohydrology of the system is simply the high amount of stem water storage, diverse rooting depths, high water use efficiency equals an exceptionally high rain use efficiency, which again defined as net primary productivity over precipitation. And we think this is especially true in saguaro dominated ecosystems. So one final thought, the reason why I think we should care about this is because if the water is removed from the soil, the good news is if you study the biodiversity of dryland ecosystems, is again, that water gets efficiently turned into productivity, which could support very diverse biota of the system. On the other hand, if you have less soil moisture, because it's actually captured in the stems, then perhaps there are other soil processes like nutrient cycling, microbial growth, etc., that actually then gets actually removed from the system and could have, in some ways, a negative impact in other particularly plant species. So that's why we think it's important to understand how these different giant cacti could impact total soil water storage and ecosystem water cycling. Thank you so much.
Thank you so much for your amazing talk, Dr. Kevin. I would like to highlight that doesn't matter from which point we're studying these plants, they are always very interesting and amazing, and it's very surprising how its adaptations to these climates just overcome many other plants. So I think we have time for some questions. Please feel free to ask them just by directly in the microphone or directly on the chat. It doesn't matter in which language, we can try to make the questions get to you. Well, if it's, I would like to start with a question. Oh, Dr. Ulises, if you can start, that would be great too.
Hi, Kevin. Really nice talk. I'm really impressed, and I have a lot of homework. I need to read your papers, but I was thinking about many things, but I just selected 3 questions. I hope it's fine for you. Number 1 is a technical question. How large do the roots have to be in order for you to be able to measure the subflow? Because I can imagine that maybe smaller roots that are very important for regeneration during after the dry season. I mean, maybe those are the most important ones for water uptake. But you are measuring, I can imagine, the larger roots, right? So number 1, how big they have to be, or what is the limit for smaller roots? Secondly, I kept on imagining every time I go to, for instance, here in Querétaro or Puebla, I keep on seeing, obviously, the soil is very dry, but the plants are flowering, the cacti are flowering, right? So I was thinking if you have been interested on linking physiological parameters to reproductive events, whether they can be predictive. You know, sometimes they start earlier, sometimes they start later, or sometimes we have observed peaks within the flowering seasons. And third, I mean, we mainly work on barrel cacti, and actually very small barrel cacti like mammillaria, but then I kept on thinking about Echinocactus platycanthus, which is also very conspicuous in Zapotitlan in Puebla. And I was thinking whether your observations, your models, can be extended to other life forms, for instance, barrel cacti, or there is a reason why you selected column cacti in particular. And now that I see, for instance, this last picture, that I see the interplay with also with prickly pears. So I was thinking about, you know, I'm sure you have thought about it, right? That this problem is even more complex than just looking at a column cacti. And thanks a lot again, and I'm looking forward to reading your papers.
Wow, great questions, Dr. Rosas. Yeah, let me start with question 1 about the roots. That's a really great question. So with that approach that we use, those probes are about 35 millimeters long. However, because of where the placing of thermocouples are, you could potentially put them on roots that are much smaller than that length. And we've put them, in fact, on some of the roots that we instrumented on organ pipe. The root diameters are more like 20 millimeters. So we can get smaller than those large roots that we showed. There's another approach too, where instead of using probes, you could apply these collars, and the collars actually heat the surface of the root. And so it's a little bit different method, but it also sort of the same concept. You apply heat and you measure where the heat gets carried away from the heat source. That approach, you can probably instrument something down to about 10 millimeters in diameter, possibly even smaller. They're a little bit more challenging to put on the plant, because they are collars and they're a little bit larger. So you need a little bit longer, straight section of root to put them on. But Indeed, you can put them on smaller roots, and I do agree with you that sometimes it's a small roots that we really, and this is especially true for the kinds of plants that you might be interested in. Mammillaria, for example, I would think would have smaller roots clearly than something like a saguaro, but I do think there's potentially an opportunity to instrument much smaller roots and those kinds of species. Yeah, second question. This is one of the key motivators for this project is understanding, can we start to link individual pulse events to how plants will respond over time? And of course, flowering is one of the most fundamental things that biologically, one of the most important things that these plants do, because they flower during a time of year, at least in saguaros, that typically these dryland ecosystems are devoid of resources. And so really important for a lot of like migrating bats, etc. So what we've found in the past, and some of my colleagues have found, that it's really hard to apply a model of what the drivers are to flowering because there's such long lag times, sometimes for several years. So for example, if you have down where you are, you probably don't have the kind of freezing events that we occasionally have. And those freezing events can really create this exceptionally long lag time of 2 to 3 years. So you could get an inhibition of flowering, and then a couple of years later, you get a flowering boom because all that energy was really not spent. But trying to link that timing of precipitation has been very difficult. What we have found in saguaros that late winter rains are really important for the amount of flowering and the timing of flowering. So rains that start like in February and March around here seem to be pretty well linked to the amount of flowering output. But those models are still a little weak in their predictive capacity. So it's been a real challenge for us up here to try to link some of the, the environmental conditions to flowering output, because there's a lot going on. And in terms of your third question about these smaller plants, including these prickly pear, I think it's really important to understand, indeed, giant cacti are kind of the extreme case because of that residence time, but we don't know what the residence time is of these smaller plants. Because it's not easy to measure, but the prickly pear and mammillaria are perhaps delaying the timing in which water is lost in the ecosystem. I think it's a really important question just because of the amount of biomass that could be on the system. My guess is that these prickly pears are cycling water much faster than these saguaros, and probably have shallower rooting depths, although I think some prickly pear plants have been shown to be fairly deeply rooted. And so that analysis needs to be done. And we have not done that yet, but I think it's really important that we can characterize residence time in relation to volume to surface area ratio, because we can measure that in these plants. And if we can do that, then I think we can start to build some predictive models.
I have a question too. I would like to ask you. I was thinking, I really don't know how closely related are saguaros and the pipe cactus, or if their niches overlap like a lot. But do you think that this divergence in the physiology arose from competence, or like if they were competing at some point and that's why they have different behaviors? Or do you think it's just a matter of the environmental factors driving that divergence in the physiology?
Well, they do have very overlapping, obviously overlapping ranges, and they do occur in similar environments. They occur you don't see Oregon pipe in the extreme northern Sonoran Desert, largely because of it's too cold, believe it or not. So they're not very frost tolerant. And then I don't know this for sure. Maybe some other folks have some better ideas, but the reason we don't see saguaros in southern Baja, I think, is because of pollinator limitations. So, but one of an interesting part of your question is, as I mentioned, that we think they have very different rooting depths. And so are those rooting depths, do those sort of differences in rooting depths allow them to sort of co-occur without direct competition in many ways for water? They do both have shallow roots, and they're both very opportunistic at taking up water after it rains. Saguaros have, if you dig up their taproot, you can see that it's basically a large storage organ where there's a lot of starch, and that allows them to build roots very quickly after a rain event. Organ pipe doesn't have that kind of storage. So it's possible that if you measure the amount of time that they start to take up water after a rain event, that I would guess that organ pipe may not be as quick to respond as saguaros, but I don't know that for sure. If you go back to look at those sap flux traces, they look pretty similar. But I think that because there are some differences in physiology, in particular below ground physiology, it does allow them to co-occur at least to a certain extent without direct competition. That's my best guess.
Well, thank you. I think we still have time for another question if someone else is interested. If not, well, again, the ICANN is very thankful for having you here, and I think we will prepare something so you can to show more about your research on social media as a present that we are very thankful for your seminar. And as commercial for the next seminar, we will have talk about saguaros demography. So we're still talking about giant cactus in the Sonoran Desert, especially. And so please keep posted on our social media. We are .ican/cactasia everywhere. So please follow us on Twitter and on Facebook, on Instagram, and also we have the past seminars on YouTube available if you missed someone or if you want to watch this again. So thank you for joining us again. Thank you, Dr. Kevin, for your amazing talk, and we hope to see you again.
Great. Well, thank you so much, Eddie. I really enjoyed it.
Thank you. Bye-bye.
Very much.
Bye, everyone.